Good morning, all. Today we are going to study the introduction to medicinal chemistry, its history in development, and I am going to take you towards the journey of how we are going to study this particular subject in our course of third year pharmacy. So, when we move further, we need to understand the subject will be called as medicinal chemistry, and after studying the subject, everybody will be able to understand and correlate the physico-chemical properties of drugs and its structure with biological action. We will be able to demonstrate the mechanism of action. The subject provides us knowledge of therapeutic uses, adverse reactions. It will help us to apply the principles of organic chemistry for synthesizing various clinically significant drugs, and it will help to reinforce the topics what we all have studied in our lower classes of first year and second year pharmacy, like anatomy, physiology, biochemistry, organic, and pharmacology. Now, the subject also has practicals, and in the theory, we have following chapters. So, around 12 chapters we are going to study. So, the topic one will be about brief introduction to QSAR, prodrug, combinatorial chemistry, and CAD. And the chapters what we are going to study about all the medicinally active agents, we are going to study the classes of drugs, including its SAR, MOA, and synthesis of the compounds. Along with it, we are going to study the nomenclature and the brand names of important marketed products with its side effects. So in the theory part, we are going to cover with anti-infective agents, wherein we have local anti-infectives, preservatives, antifungals, UTI agents, anti-tubercular agents, antiviral, anti-AIDS, anti-protozoal, anthelmintics, anti-scabies, and anti-pedicular. It goes on next with sulfonamides and sulfones, then with anti-malarials, then a huge lengthy topic of antibiotics, followed by anti-neoplastics. Then we have one particular category of drugs which act on cardiovascular system, wherein we are going to study antihypertensives, anti-anginals, anti-arrhythmic, anti-hyperlipidemic, coagulants and anticoagulants, and the drugs which are acting on the endocrine system. We are also going to study hypoglycemic agents, thyroids, antithyroid agents, diuretics, diagnostic agents, and steroidal hormones and adrenocorticoids. This will be the part of the theory what we are going to study. In practicals, we'll be having practical for 3 hours in a week, wherein we are going to perform assay of important drugs from the content which is being given. We are going to also prepare medicinally important compounds or intermediates which are required for synthesis. We are going to study the monographs of few important drugs, and we are going to determine the partition coefficients, dissociation constants, and molar refractivity of compounds for qualitative structural activity relationship analysis. Now, to understand medicinal chemistry and medicine, we need to understand the difference between a drug and a medicine. So drug is any substance presented for treating, curing, or preventing disease in human beings or in animals, and it may be used for making a medical diagnosis or for restoring, correcting, or modifying physiological functions. A medication will be defined or will be referred as a pharmaceutical dosage form or simply a drug, which is used to diagnose, cure, treat, or prevent the disease. A medicine is a substance taken to cure and improve the symptoms of an illness, and medicines are used as preventive medicines which are being dispensed by the pharmacist by various regulations, right? So if we want to differentiate between drug and a medicine, in simple terms, drugs are chemical substances which are used to cure, and a medicine is a compound in one particular formulation which is being prescribed by the physician and is being dispensed by the pharmacist to the patients. So, when we want to see what are the types of drugs which are being dispensed, so we have OTC, we have prescription only medicine, and we have BTC. Further, the drugs are also classified or termed as medicine, recreational drugs, and are called as nootropics, right? So substances that affect the CNS and are used for perceiving beneficial effects on perception, consciousness, and personality are the drugs which wherein the examples are opioids and hallucinogens, diphenhydramines. Nootropics are the drugs that are claimed to improve human cognitive abilities, and they are used to improve memory concentrations, thoughts, mood learning. Some are used to treat the neurodegenerative diseases, mainly the Parkinson's disease and the Alzheimer's disease. Next, we will be studying what chemistry means. So chemistry is known as the central of science. It is a branch of physical science that studies the composition, structure, properties, and changes of matter. And if we are going to see the spelling of chemistry, each word we can quote a meaning of the word. So C stands for community, H for health, E for environment, M for medicine, I industry, S science, T teaching, and R is the research. And Y is the student who learn this, right? So there are three branches wherein we can bifurcate chemistry. One is physical, second is inorganic, and third is organic. So physical is the branch of chemistry concerned with application of the techniques and theories of physics to study the chemical systems, chemical kinetics, electrochemistry, spectroscopy, photochemistry. Inorganic deals with synthesis of behavior of inorganic and organometallic compounds, and the branches are bioinorganic, cluster, material, nuclear chemistry. Organic chemistry studies the structures, properties, and reactions of organic compounds and organic materials, and the allied branches are biochemistry, biophysics, bioorganic, and pharmaceutical medicinal chemistry. Now coming to our subject and the definition. So it is a discipline or intersection of chemistry, especially synthetic organic and pharmacology. And we define medicinal chemistry as a chemistry-based discipline which involves aspects of biological, medical, and pharmaceutical sciences. So medicinal chemistry involves discovery, development, identification, and interpretation of mode of action of biologically active compounds at molecular level. It is concerned with invention, discovery, design, identification, and preparation of biologically active compounds, the study of their metabolism, the interpretation of mechanism of action, and construction of SARs. Now, when we study medicinal chemistry, we need to understand three different stages wherein medicinal chemistry and the discoveries takes place. So medicinal chemistry stage 1 is discovery stage, which involves choice of therapeutic targets, an identification or discovery and production of new active substances, an optimization stage, and a development stage. This particular slide explains us about various disciplines which involve the allied subjects of organic medicinal, biochemistry, biology, pharmacology, toxicology, and the biopharmaceutics approach. So medicinal chemistry is always interlinked with the substances which we see, but it is not only the sole field. We need to understand spectroscopic approaches, the CAD approaches, the combinatorial chemistry, the computational aspect, molecular docking, of course, with the basics of the organic chemistry, which then, along with pharmacology, immunology, and genomics, goes on to the discovery. Now, when it has come to stage 1, we need to find a lead. So to finding a lead, we need to choose a disease, choose a drug target, need to identify a bioassay, find a lead compound, isolate and purify the lead compound, and determine the structure of the lead compound if necessary. When it comes to drug development and drug design, so first we will see drug design. If we identify the SAR and the pharmacophores, we improve the pharmacodynamics and the kinetics, and in drug development, we patent the drug, carry out preclinical trials to study metabolism, toxicology, and stability study, and design a manufacturing process and chemical process for the development. We would carry out the clinical trials and then register the drug into the market for the patients. Now, to understand medicinal chemistry, we need to understand the definition of structural activity relationship. So it is the relationship between chemical structure and pharmacological activity for a series of compounds. Lead compound is a compound that has a desirable biological activity with therapeutic relevance, but typically has some shortcomings that is likely to be overcome through the development of analogs. Agonist is an endogenous substance or a drug that can interact with a receptor and initiate a physiological or a pharmacological response. An antagonist is a drug or a compound that opposes the receptor-associated responses normally induced by another bioactive agent. Now, why there was need to develop medicines. So it is going to improve the lifespan, and it is going to increase the life expectancy. So if we see the examples like wherein all the vaccines were being given, so bacterial and viral infections, so medicines are used for them. Medicines are used to control the birth. Medicines are used for the life threatening diseases like cancer, HIV, and malaria. Now, pure organic compounds are the compounds or the agents which are used to cure, mitigate, or to prevent the diseases. So these remedial agents are classified based on the origin. So drugs are being classified based on from where we get them or where we isolate. So we have natural, we have synthetic, we have semi-synthetic. These compounds are basically based on the literature which is available in the Materia Medica in 2000 BC, wherein 250 vegetable drugs and 120 mineral drugs were being isolated. Further, it went to 1500 BC, wherein Egyptians gave the literature about 700 drugs originated from animals, plants, and minerals. Later, the Emperor Frederick issued the Magna Carta of Pharmacy in 1240, further which evolved the synthesis of urea in 1828, which started medicinal chemistry. And then the medicinal chemistry received formal recognition in academic pharmacy in 1932. Now, when we talked about the development and the evolution of medicinal chemistry since 2000 BC to 1932, we need to also to understand how these things came into the picture and how the market was evolved. So the literature tells about types of medicines from their origin. So we have Chinese medicines like Chinese herbs, some of the ingredients which are still in use, mainly reserpine, which is going to be used to treat high blood pressure, emotional and mental control, and the ephedrine, which is used in asthma. Egyptian medicines, Papyrus Ebers, he has given 877 descriptors and recipes for this. Greek medicine from 700 BC, which was being conveyed to the world that illness is not a punishment. Medicine is considered as science. So diseases are due to natural causes, and hence the evolution of medicines came into the picture. Then Romans developed the field, and wherein invention of hospitals, large influence of Greek medicines, and the introduction to materia medica, the pharmaceutical descriptors were being given to the market. The era of horrible epidemics wherein leprosy, chickenpox, tuberculosis was there. So therein various methods were developed by the scientists, and development of scientific approaches. To quote a few examples, pox was being developed, wherein Edward Jenner discovered that people who worked with cattle and had caught the cowpox disease, a mild disease related to smallpox, were immune and never caught smallpox. He inoculated a baby boy with a blister fluid from a woman with cowpox, and later inoculated the same boy with fluid from smallpox, and discovered that the boy was immune against the disease. Bill Withering introduced extracts of digitalis for treatment of heart problems, and Louis Pasteur discovered microorganisms which are responsible for diseases and developed vaccinations against rabies, and he introduced the attenuated viruses for treatment of rabies. Digitalis, quinine, ipecacuanha, aspirin, and mercury are the agents which are used to treat several of the disorders. Alexander Fleming in 1928 gave the fungus information, wherein biotechnological products of penicillins were evolved. And Robert Koch was being given a Nobel laureate for discovery and treatment of tuberculosis. Early 1900s, synthetic drugs, foundation of pharmaceutical industry took place. Later in 1930s, screening of natural products, isolation of bioactive ingredients. Later in 70s, development of recombinant drugs, proteins, interferons, and development of biotechnology. Further evolved in 2000, deciphering of human genome therapy, investigation of the molecular basis of disease, and currently the personalized medicine era is going on. Now, present medicinal chemistry, gene therapy, wherein technique for correcting defective genes, Here it is the process of inserting genes into cells to treat the diseases, and gene therapy is used to correct a deficient phenotype. Gene therapy vectors, wherein viruses are being used, to name few: adeno associated and herpes simplex virus, wherein pure DNA constructs were being done by making the molecular conjugates which were intercalated with the DNAs and human artificial chromosomes. Now, this particular slide tells about the history of development of medicinal chemistry, chronology of drug innovation, wherein from 1890 to 2010, the drugs were evolved and the information was for the benefit of the humans for the treatment of various diseases. Development of the classes took place further. So during 1840s, the first use of synthetic organic chemicals were introduced for anesthesia during a tooth removal, such as nitrous oxide, ether, and chloroform. In 1864, barbituric acid has been synthesized as a useful hypnotic agent. In 1875, salicylic acid was introduced as a possible cure for typhoid fever, which had an effective antipyretic action also. Later in 1899, aspirin was marketed as an antipyretic without any of the unpleasant side effect, and this indicated that chemical structures from natural products were changed into better drugs. This particular examples tell us the evolution of the drugs. First is salicylic acid. Later, and salon like phenyl salicylate was prepared, which has shown an improved solubility parameter and was better tolerated. Hoffman in 1890 at Bayer's tested acetyl salicylic acid and was with a good advantage, and now this is being used widely and it is being rapidly hydrolyzed in the gut to give active salicylic acid. It is the example of a prodrug which is in the market. Finazone was synthesized in 1884 and was the most popular drug used after aspirin and was used as an antipyretic. Similarly, in 1853, modifications were done on the naturally occurring morphine, wherein quaternary ammonium salts and the substitutions by HNCH3, namely in morphine and codeine, improved the efficacy of the drugs in the market. 1910 and 1920s and 30s, this particular development led to the functional group modifications of the pharmacophores, and which was investigated. So combination therapy was being into the use, and theory of antimetabolites was postulated, wherein the metabolic products were used as lead compounds to treat cancer. In 1950s, aging diseases, cerebrovascular and cardiovascular, became first reason for human death. New drug design based on enzymes and receptors was taken place as new targets. In 1960s, oral steroidal contraceptive agents were discovered, and corticosteroids have become an important drug. Later in 64, beta adrenergic blockers were introduced into the market. Propranolol is the example. Later, nifedipine and calcium channel blockers were marketed. In 1981, captopril and ACE inhibitors were launched. This particular slide explains us about the pre and post era in the development of drugs and in the history of drug discovery, where in 1990 gave us herbal drugs, serendipitous discoveries, 20s and 30s vitamins and vaccines, 40s antibiotics, 70s biotechnology was arose, 60s there was breakthrough in the etiology, 1950s new technologies, discovery of DNA, 80s commercialization of drug discovery, combinator approaches, and 1990s, and now in the decade of 2000, we have robotics, automation, and AI in drug discovery. This particular slide explains about the drug discovery and development timeline, wherein it is a funnel which is being inverted, and there are 10,000 of the compounds which were thought of studying further, which wherein in preclinical trials there were 250. Further in clinical trials, 5 compounds come. It is just an overview to show you all how the development goes on, and then further the drug is being approved by the FDA, and one FDA drug approved comes to the market for its therapeutic use. Drug discovery process involves the following stages. So first is target selection, wherein cellular genetic targets are selected, wherein by genomics, proteomics, and bioinformatics these areas are being used. Lead discovery, wherein we synthesize and isolate the molecules, and the approaches used are combinatorial chemistry, assay development, HTS, high throughput screening. Medicinal chemistry involves the development of the libraries of the drug molecules, the structural activity studies, the in silico screening, and the synthesis part. In vitro studies after synthesizing, wherein drug affinity and selectivity are studied. Cell disease models are studied to identify the mechanism of action, and the lead candidate is being refined. In vivo studies helps in modeling of the drugs and studying the animal models for the disease state. It's behavioral studies, it's functional imaging, and performing the ex vivo studies, further which involves clinical trials and the therapeutics later on. So drug targets and the development is based on three main approaches. One is mechanism-based drug design, second is structure-based design, and third is receptor-based design. With this, I have made the presentation in a very lucid manner wherein students can understand the importance, the definitions of medicinal chemistry, the basic terminologies which we use, with the approaches which are used to study medicinal chemistry, and the drug development, of course, the history part, which is very, very important to study. Now, how medicinal chemistry works hand in hand with the other allied disciplines. So we have the collection of stored chemicals along with the associated database, which assist in high throughput screening, which helps in the drug targeting based on the basics of organic chemistry. Further, SAR studies, which helps to identify the pharmacophore in a precise manner, which is responsible for activity, and further it enables to prepare more active compounds by eliminating the existing side effects of the drug. Further, in silico screening helps the simulation studies to find the structures and the most likely binded drug targets. Further, with the approach of synthesis, we can synthesize them. Few of them later, when they are synthesized, they are characterized, and then in vitro and in vivo studies are being performed for them, which leads to phase 1, phase 0, 1, 2, and 3 in the clinical trials, and then submission of the new drug application in the formal step is being given to the FDA to consider a drug for marketing approvals. FDA reviews, approves the applications, and after the approval, phase 4 studies, that is post-patient surveillance is being done, and the drug exhibits into the market. So this is all to explain about what medicinal chemistry is in the course of PharmD, how it evolved, the history of medicinal chemistry. The topic helped us to understand the time frame of the discovery of the molecules till it comes to the clinical development into the market for the use. So thank you for today, and we are going to start with the next topic tomorrow, wherein I'll be starting with the anti-infective agents.